Welcome to the fascinating world of chordates. Imagine you are trying to build a tall, sturdy skyscraper. To keep it from collapsing under its own weight, you need a strong central steel pillar running right down the middle. In the animal kingdom, nature faced a very similar problem when trying to build larger, faster, and more complex creatures. Today, we are going to explore how nature solved this problem, defining an exclusive club of animals that includes the birds in the sky, the fish in the sea, and even you. Building on that skyscraper analogy, let us look at how nature actually built that central pillar. Nature needed a way to support larger and more complex creatures without them collapsing. Instead of a rigid steel pillar, nature invented something called a notochord. This acts like a flexible biological scaffolding. Animals that possess this special scaffolding, along with a few other specific features, belong to an exclusive club called the Chordates. By understanding how this club is defined, we can trace the family tree of the most complex animals on Earth. Now that we know what a notochord is, let us define the exact membership rules for this exclusive club. In biological terms, the phylum Chordata is defined by four distinct anatomical hallmarks. First, they possess the notochord for skeletal support. Second, they have a dorsal hollow nerve cord, which eventually develops into the brain and spinal cord in higher animals. Third, they feature pharyngeal slits in the throat region, modified into gills or jaw structures. Finally, they have a muscular post-anal tail. Even if an adult human does not visibly show all these, they were absolutely present during embryonic development. Before we move on to why these features matter, let us see this with a concrete visual example. Here, we are looking at a generalized chordate body plan. Notice the long pink rod running down the back? That is the notochord, providing structural support. Right above it, the blue tube is the dorsal hollow nerve cord, the future nervous system. Towards the front, you can see the pharyngeal slits in the throat area. And finally, look at the back end, where the muscular post-anal tail extends clearly beyond the digestive tract. This simple blueprint is the foundation for all vertebrate life. Having seen the blueprint, you might wonder why we study these specific traits so closely. Understanding the defining features of chordates is absolutely crucial because it provides the evolutionary roadmap for our own existence. Every major medical breakthrough involving animal models relies on the shared biological architecture we have with other chordates. Without this connection, we could not use mice or zebrafish to study human diseases. Furthermore, recognizing these traits helps conservationists track the health of ecosystems, as chordates often serve as apex predators. This knowledge bridges the gap between simple marine organisms and complex mammals. Now that we understand the biological importance of chordates, let us look at how we measure their presence in the wild. To truly appreciate the diversity of chordates, biologists use mathematical models to measure how varied a population is within a specific habitat. Biology is not just about memorizing body parts. It is also about quantifying life. We need to know not only how many different species live in an area, but also how evenly distributed their populations are. A healthy ecosystem requires a good balance, and we need a specific tool to calculate that balance. This need to measure balance leads us to one of the most common tools used in ecology, the Shannon-Weiner Diversity Index. Conceptually, this formula captures both the richness and the evenness of species in a habitat. Let us break down the symbols. H represents the Shannon Diversity Index itself. S stands for the total number of different chordate species present. The variable p sub I represents the proportion of individuals belonging to a specific species. We multiply that proportion by its natural logarithm. When the number of species increases, or when populations become more evenly balanced, the value of H increases, indicating a healthier ecosystem. Now that we have our formula, let us see this with a concrete example. A biologist is studying a small pond ecosystem and counts the chordates present. She finds 50 fish, 30 frogs, and 20 turtles. We are asked to find the baseline health of this community. First, we must find the total number of individuals, which is exactly 100. Next, we calculate the proportion for each group by dividing their count by the total. The fish make up 50%, or 0.50. The frogs are 0.30, and the turtles are 0.20. These fractions are our p-values. With our proportions calculated, we can now plug them into the Shannon Diversity Index formula. We take each proportion, multiply it by its natural logarithm, and add those products together. Because the natural log of any fraction is a negative number, the sum inside the parentheses will be negative. That is exactly why the formula has a negative sign at the very front. It converts our final answer into a positive number. After doing the math, we get a final index of 1.029. This positive value gives us a baseline metric for the pond's chordate diversity. To truly understand what that index number means, let us compare two different ecosystems. A researcher wants to compare Forest A and Forest B. Forest A has 80 birds and 20 mammals. The total is 100 animals, so our proportions are 0.80 and 0.20. Notice that this population is heavily skewed toward birds. When we plug these skewed proportions into our formula, we calculate a diversity index for Forest A of 0.500. This relatively low number reflects the lack of balance between the two chordate groups. Now, let us look at the second forest in our comparison. Forest B also has exactly 100 animals, but it has 50 birds and 50 mammals. The populations are perfectly even. Our proportions are 0.50 for both groups. We substitute these beautifully balanced proportions into our diversity equation. Multiplying each by its natural log and summing them up, we get a final diversity index for Forest B of 0.692. We now have two distinct mathematical results for two forests with the exact same number of animals. So we have calculated the indices for both forests. This brings us to a formal proof of a key ecological principle. We see that the index for Forest B, 0.692, is strictly greater than the index for Forest A, 0.500. Remember, both forests had the exact same total number of animals and the exact same number of species groups. The mathematical reasoning here isolates a single variable, the evenness of the population. Because the balanced forest yielded a higher index, this mathematically proves that ecosystems with an even distribution of species are more diverse and resilient. To wrap up our journey, let us review the key takeaways. First, the phylum Chordata is united by four incredible evolutionary innovations: the notochord, the dorsal hollow nerve cord, pharyngeal slits, and a post-anal tail. These features provided the biological foundation for the evolution of highly active animals, including ourselves. Second, we learned that biology relies on mathematics. By using tools like the Shannon Diversity Index, we can actively measure the health of these fascinating creatures across different ecosystems. Mastering these concepts is your first major step into the incredible world of vertebrate zoology.